So it is now my great pleasure to introduce Professor David Nutt. I'm sure that he does not need an introduction, but I will try to do one anyway. He has many different hats that he wears, from his role in politics, in academia, research, and psychiatry. And I think perhaps the reason that we are all here this evening, due to his wonderful work as an educator through the several books that he's already published, his founding of drug science, and the really amazing amount of brilliant free resources that you can access there. And of course, his many public lectures, which I believe is the birth of this wonderful book that some of you may have been hearing that I've been speed reading recently. The Brain and Mind Made Simple. I think this started in a series of very popular lectures that David was giving pre-pandemic, if we can remember such a time. And the book takes you through from the beginning of, understanding chemical signaling in the brain, right through how that affects different pathological states and our different experiences as people in the mind. And I won't tell you any more than that, and I will hand over to David.
Well, it's great to be giving this lecture. So the lecture originally started off like this. It started off as Brain in the Mind Without the Hot Air. And the plan was that this would become a follow-up to the drugs without the hot air. But the publisher said that he didn't think there was a market for a brain book, or not the way I'd written it. So I just hope he's wrong, because the book is out there, and hopefully some of you will buy it and some of you will like it. So I changed the title, of course, in a fit of pique. Well, in fact, I couldn't use hot air because that's the UIT publishers. That's part of their copyright. So I changed to Made Simple. But this is the origin. And these lectures I gave for drug science 3-4 years ago, and they were surprisingly popular. So I thought, well, why not turn them into a book and see? So this is the lecture. I've condensed it down a little bit, so we've got more time for questions. So it's a 45-minute talk. I've taken out a few of the slides, but I start in the place where we all started, of course, which we all started as bacteria. We all started as these unicellular organisms, which were probably self-assembled through some combination of chemical interactions in the primordial soup. Although, of course, they could have been seeded from another planet that's older than ours. But the important point, and the reason all life is based on a very simple principle, which is that you have a cell membrane. So bacteria are cells, they have a single cells, they have a membrane, and the cells survive because they have pumps, and pumps are central to all life, and they're very central to how nerves work as well. And the original pump was a pump that actually squirted sodium bacterium to the outside into the seawater in which they were living. And all cells, pretty much all cells today that we know of, do the same thing. They pump out sodium, and that allows the inside of the cell to work, because, as you kind of know, almost certainly, putting things into a strong salt solution kills them. We use it as a disinfectant, we preserve foods, salt cod, etc. And so for life to work inside a cell, you've got to get the sodium out. And it turns out that that's important because, particularly in nerve cells, reversing that pump to let sodium come into the cell is actually how nerve cells work. I'm not going to talk more about that tonight to save time, but I'm going to talk about the development of the nervous system. And it goes back a long way. This is one of the most simple organisms with a nervous system. This is the Hydra. It's named because it's got these snake-like tentacles that come out of the head, like the ancient Greek mythological creature. And Hydra have a primitive nervous system, and they really highlight actually what the first purpose of the nervous system was, which is to move tentacles around to search for food. And you can see that the reason we have a nervous system, all creatures that have nervous systems look for food, and they're called animals. And creatures that don't really have a nervous system are called plants, and they don't need to look for food because they make food. So some people argue the nervous system is all about hunting for food. And certainly it plays an enormous role in even very highly evolved animals hunting for food like humans. And sometimes, of course, we take in too much. But we shouldn't be too we shouldn't be too dismissive of plants. Some plants do have kind of primitive nervous systems. They can react to stimuli. They can react by closing their leaves, like the Venus. The principles of moving a plant leaf are very similar to the principles of moving a limb or hydra, flagella. Ions move to change contractility. And after movement, the next thing that happened was detection. So hydra detect food through chemosensors, which pick up sense chemicals that are produced by animals, plankton that float by, and they can then move to collect them. And of course, one of the most powerful, maybe for humans, the most powerful senses of all are the eyes. And eyes have been around for a long time. Even jellyfish have eyes. There you see some eyes on a jellyfish. So light sensors have evolved multiple times. The eye itself has evolved multiple times in the course of evolution, because detecting things by, through the light emission is also very important. And some creatures can detect light we can't see. So the cobra, for instance, has under its in between its eyes, it has a special pit that detects infrared light it knows exactly where humans are or other animals are by the heat they radiate. So you can have see outside the visual spectrum.
David,
really important. Yes.
Sorry, we're struggling a little bit. I think the Wi-Fi's playing up a bit. Do you want to maybe try if you take your video off? That might help.
I will certainly do that. So let's get. Yes, it says I'm unstable.
Sorry.
So let's no problem. Thanks for letting me know. You don't need to see me. So hopefully I sound a bit better now. Do I sound better?
Yeah, much better. That sounds good.
Great. Okay. I'll let you see me at the end again, just to remind you I'm still here. So the real breakthrough that came in terms of developing a complex nervous system was the way in which neurons learn to communicate with each other. And they do that almost always through the release of chemicals, which we call neurotransmitters. So this is a very simple diagram, but it covers the fundamental principles. The brain is a chemical organ because most of the communication is done through the release of these neurotransmitter chemicals, which cross a thing called the synapse, which work on postsynaptic receptors, and those receptors convey information to the postsynaptic neuron. And there are one or two fundamental aspects of this synaptic organization. One is that there are uptake sites. So for most neurotransmitters are stored in vesicles, they're released into the synapse, they're taken back up through transporters or reuptake sites, and then they're recycled. And this is an efficient way of maintaining or minimizing the energy to produce neurotransmitters. And there are receptors on the receiving neuron, and also what we call autoreceptors on the presynaptic axons, which also regulate activity. So it's a sophisticated network. And also, since we've recently discovered the endocannabinoid system, we realize it actually, you don't just have receptors on the postsynaptic neuron, you also have activation of these endocannabinoids, which float back across to work on the presynaptic neuron to often produce another kind of feedback. So it's a very balanced system: release, feedback, release, feedback. Now, in simple terms, there are two kinds of neurotransmitters. There are the fast-acting ones, which are the on-offs, which are the brain. And for the purposes of our brain, we're talking just about two amino acids. One's called glutamate, which is excitatory, and one's called GABA, which is inhibitory. And everything that you learn from my talk today, or from any other talk you've ever listened to, or any other thing that's laid down as a memory is laid down because of glutamate. Glutamate is the on switch. but it's very powerful, and therefore GABA is always around with it as an off switch, just to make sure it doesn't get out of control and you don't overexcite the brain. And then there are these slower acting neurotransmitters we call neuromodulators, and they comprise actually a very small proportion of all neurons, maybe about 5% of all neurons. And these are transmitters like dopamine, serotonin, noradrenaline, and they're also peptides like endorphins. And they have an important role in adding what we call valence to the main impact of glutamate and GABA. So they're involved in emotional regulation, emotional memory, and drives, and adding, as I say, valence to memories. And we'll come to those later. So the very simplest explanation of how the brain works is that glutamate is released by neurons, and these neurons are called pyramidal cells, and there's a pyramidal cell, that's what they look like. And they have one axon that shoots out and then branches and can activate up to 1,000 other neurons. And they release glutamate, and glutamate is excitatory. It stimulates the postsynaptic neuron. In all mammals, glutamate turns on. It's the on switch of neurons. And it lays down memories. It's what's responsible for all learning. But if there's too much glutamate, you can get anxious. And if you have too much glutamate, you have seizures. And glutamate is a major transmitter causing epilepsy. So because glutamate excess is very damaging to the brain, every time glutamate is released, there's always the stimulation in parallel of GABA. A GABA interneuron is shown here. So the GABA neuron is stimulated. That also releases a transmitter, and that transmitter is called GABA, and GABA calms the brain. And so if GABA is responsible for sedation, for calming, for muscle relaxation. And of course, if you have too much GABA, which you can do if you, for instance, if you drink a lot of alcohol, which mimics GABA, then you become unsteady and you can fall over. So GABA calms, glutamate excites, and they work in a balance. And one way of thinking about the brain is it's like the brain is walking a tightrope, always between too much glutamate and too much GABA. So this is the way I conceptualize the GABA glutamate axis. It's the axis which is the regulates what you might call normal consciousness. So currently now you're all hopefully aroused, paying attention, you're laying down memory, and that's because glutamate is dominating in relation to the information that's going into your brain. And one way of thinking about the power of glutamate is concept of parcellation. The ridiculous precision that you have in terms of the definitions of your visual, your auditory, and your memories, and your also in terms of the density of information that you remember. This precision with which you segregate things into your brain is parcellation, and this is down to the effect of glutamate laying down memories. And if the GABA system becomes overactive, then you fall asleep and go to coma, and that of course is what happens in anesthesia, and we induce that deliberately to avoid having memories, for instance, of having a major surgery. So a little bit more detail as to how these neurons work. Well, here's an example. So this is a schematic of all these hundreds of millions, maybe estimates vary, but up to 160 billion pyramidal cells make up the brain. So your brain is largely a forest, a really big forest of pyramidal neurons. And they have these dendrites which come up, and the apical dendrites which branch and get input from other pyramidal cells. So you can see you got over 100 billion of these, and each connects to 1,000. So you've got trillions of connections in the brain. And then you have these GABA interneurons which keep the control to make sure that the brain doesn't overstimulate it and have a seizure. And here you see the stimulation of pyramidal cells, which these pyramidal cells are being powerfully activated by stimulating them. And here's a schematic of what a pyramidal cell looks like. This is the pyramidal bit where the cell body is. Here's the axon that goes out and spreads out to, say, 1,000 other cells. And then the receivers of the pyramidal cells are these dendrites, as we call them. And these dendrites have branches. They're like a tree. They're called arborizations. And on the branches, you have these things called synaptic spines. So you have little protuberances into which you get inputs like here from 1,000 other pyramidal cells. And each pyramidal cell is like a computer. It's got a computing power that's pretty close to that of a literally a small computer. And that's why the brain is an amazing computer, because you've got 160 billion computers that all connect to each other to make a supercomputer. And the brain has more computing power than all the computers on Earth put together. That's been estimated, even for a mouse brain. So a human brain is much greater. Now, those pyramidal cells, because they depolarize and they have electrical impulses which travel down them to the next cell, they produce massive changes in electric activity in the brain. And we can measure that using things like this technique called EEG, electroencephalography. And here you see the different changes in electric activity measured across the different electrodes that are placed on the skull. And EEG measures electric activity. And that electric activity changes during the different stages of sleep. And one of the great advances that have been developed as using EEG is to allow us to measure sleep stages and monitor sleep stages using a technique we call polysomnography. And poly means measuring other things. So you can't exactly quantify or define the stage of sleep people are in using EEG, because when you go into dreaming sleep, it looks like you're awake, which is why dreaming sleep used to be called paradoxical sleep. But using a mixture of EEG measures and measures of eye movements, we now call dreaming sleep rapid eye movement, REM sleep, and also myographic activity in the submental muscles. In dreaming sleep, you get paralyzed muscles. By using this mixture of EEG and other measures, we can quantify this and measure the different stages of sleep through the night. And this is what you can use this technique to produce what's called a hypnogram. So this is a measure of the different stages of sleep in a typical night. So here you're all awake, and then when you go to bed tonight, you will have a little period of being awake. Of course, after an exciting talk like mine, you might be awake for hours thinking about it, but probably not. But when you drop off to sleep, you'll go into stage 1 and stage 2, the very light sleep from which you're easily aroused. And then you'll go into deep sleep, stage 3 and 4. This deep sleep is the restorative phase of sleep. And that lasts, if you're lucky, you're young and in your 20s or 30s, you might have maybe an hour of that. And then you'll come out of it and you'll go into the first phase of dreaming sleep. And then you'll have another cycle. You go back into deep sleep and then dreaming sleep, deep sleep, dreaming sleep. And you'll see a fundamental phenomenon of sleep, which is that in the beginning of the night, you get a lot of deep sleep and less dreaming sleep. And in the end of the night, you get a lot of dreaming sleep and not very little deep sleep. And the reason for that is that this is a restorative. This phase of sleep is where you start secreting hormones like growth hormone. This is the sleep you catch up on if you ever sleep deprived. This is probably the best kind of sleep you can have. And dreaming sleep, I'm not entirely sure what dreaming sleep does, but it's certainly, if you sleep deprived people, you get a lot more of the deep sleep before you get more of the dreaming sleep. You'll also notice that people wake up during the night, and you have to be awake often for most people, more than 2 minutes to actually be aware you're awake, or you have to look at the clock and get angry with the fact you're awake. For most people, there are multiple awakenings. And as you get older, the deep sleep gets less and less and less. So at my age now, I don't have really any deep sleep, but I still have dreaming sleep and I have a lot of awakenings. And I just want to say a word about alcohol, because I've already touched on the fact that it potentiates the effects of GABA. So this is the GABA glutamate balance I've talked about before. And alcohol enhances GABA, which is why it relaxes you, makes you feel more sociable. But that's after the first 1 or 2 drinks. But if you carry on drinking, then alcohol has another effect, which is to block glutamate. And it's when you get to that point, you get into what's called a blackout, because if you block glutamate, you can't lay down new memories. So then people forget. They have patches of the night when the glutamate system is so blocked, they can't remember. And that blockade of glutamate leads to other factors. It can, in excess, it will kill you, because if you don't have any electrical activity in the brain, then you can't breathe. But it also, this is an important, the blockade of glutamate receptors, it leads to neuroadaptive changes in the brain, which then lead to withdrawal, and probably also contribute to the craving and the dependence on alcohol. And I'll just give a plug for one of my other books, which was out a while back, Drink, which is a perfect companion to the brain book. And this is a particularly important one for anyone that does drink too much. Give it to that person. Now, I want to focus a little bit now on how we measure brain activity other than using EEG. And the technique which I'll share with you throughout the rest of the talk, one of the most important techniques is this technique called magnetic resonance imaging, or functional magnetic resonance imaging. So it uses a scanner. The scanner basically is a circular magnet which has a magnetic power 1,000,000 times stronger than the Earth's magnetic field. And it's so powerful that it can align hydrogen atoms in water, and changes in water flow in the brain, and changes in oxygen saturation in the blood flow to the brain can be picked up by using this fMRI technique. And here's an example of our research looking at the emotional system in the brain. So looking at, if I was to show you these images, which you've now looked at, images of horror, very unpleasant car crash, an image of someone threatening you with a knife, before you even realize what you were seeing, this part of the brain, the amygdala, would become active. So this image here shows the increased blood flow, which is a measure of brain activity in the amygdala in response to these very images. And the amygdala is a very important detector of threat. And the amygdala will start you avoiding or running away from threat even before you know you've actually seen something threatening. And this is a system which we can activate using these images, and it's a way of we can measure the activity of this system and show it's altered in conditions like PTSD, for instance. But you'll also notice the visual cortex is also massively activated by these threatening images, much more so than by ordinary images. If we just showing you a picture of a tree or flowers, you wouldn't get this activation. So the emotional response to a visual stimuli is encoded both in the amygdala and also the visual cortex, but this is subconscious activation. And you're probably aware that there's an enormous amount of research in terms of brain science using the mouse. And I think whilst that's very good science, it isn't necessarily directly applicable to the human. And I wanted just you to show the comparative size of a mouse brain and a human brain. And I don't think any of us are going to argue that the human brain has got a much greater capacity than the mouse brain, although in principle, the human brain is really just a much, much bigger mouse brain. The fundamentals of brain function are present in the mouse as well as the human, but perhaps not the sophistication, particularly in conditions like self-awareness. But we know in dolphin brains, and we know in elephant brains, in gorilla brains, that these brains are large enough to have self-awareness. Dogs and cats don't really have that. So there is something about the brain size which eventually adds dimensions of functionality, which are particularly relevant to things like insight and self-awareness. Theory of evolution of the brain, which really says there are 3 brains within one. So in our brain, we have the reptilian brain, which is the brain that you'd see in a frog or a newt, a salamander. And it's a brain that's actually pretty efficient. It can do a lot of things. It can move, it can eat, it can reproduce, it can take evasive action, and it can learn. And then building on top of that, we have another level of sophistication, which is called the paleo-mammalian brain, and that's where at the beginnings of the emotional system. This is where you begin to have the development of emotions with anticipatory anxiety, fear responses, etc. And then on top of that, you have the mammalian brain with this massive expansion of the cortex, which is particularly evident in humans, and as I showed you in dolphins and elephants. And it's in this high level of cortical activity that you get the high level cognitive, linguistic, and self-referential processing of the human brain. And those three levels actually turn out to be functionally relevant. It's not just that they're anatomically or evolutionarily relevant, but they're actually functionally relevant. So this is what I've just told you. You go from the frog brain, the pre-mammalian brain, to the dog brain, where you have this massive expansion in terms of emotional responsive, but also affiliation and bonding. The dogs are extraordinarily powerful in terms of developing relationships with other dogs, and of course with humans, and being very and living in communities. And then as you get up to the human brain, where we have the higher levels of thinking, feeling, planning, language, and self-awareness, and what we know is that activation through this pathway is really important in terms of driving activity. And therefore we have at all levels reciprocal inhibition. So what we call this top-down control, that the cortex, the high levels of the cortex, regulate this limbic system, and the limbic system regulates the hypothalamus and the brain stem, where these primary functions occur. And this top-down control is a fundamental feedback loop, which you can see in massive amounts of human behavior, and but also in animal behavior. And I just want to show you this. This is top-down control in action. So these are my three Labradors, and they've got food there. You can see their food, and they're desperate to eat. So their hypothalamus and their subcortical areas are saying, eat, eat, eat. They are desperate to eat. But I've trained them not to eat until I tell them they can eat. And that control is coming from their cortex. So their cortex is saying, I must not eat. I must do what I'm told. But as soon as I give them the cue to eat, they will eat. So you can see that there's an enormous pressure to eat, but they're restraining it.
You may have it.
And that's just a rather vivid example, a real example of top-down control. They can control their behavior in a way it's actually pretty impressive. There are plenty of humans who probably couldn't do it as well as they do. So this top-down control is important for regulating much of human behavior, but it's also important in that there are disorders where the top-down control can be excessive, and disorders like anorexia, where people won't eat, and OCD, where people will control their behavior excessively. They're examples of where the cortex essentially over controls the rest of the brain, and they're problematic in terms of how breaking these overlearned behaviors can be very challenging. Now, of course, the brain is enormous. Human brain is enormous, and it's very sophisticated, and it's not just a single homogeneous function. It's homogeneous in the sense that all the different bits of the brain look the same largely, but they do different things. And this is one model of what the brain does, which is not quite right. It's got some important constructs in there, but the location's wrong. We now know from previously from brain injury studies and cancer studies, and now from imaging studies, that the different parts of the brain do different things. So the visual system is here in the back, and the planning control systems at the front, and the auditory systems and the temporal lobes, etc. One of the more interesting things we've discovered, which I'll come back to later in my talk, through brain imaging, is that there are centers which also control what you might call aberrant emotions. And it turns out there's a cortical center for depression, isn't it? And it's this area here. This is the front of the brain, back of the brain, cerebellum here. And this area here, which is part of the high level cortical system called the anterior cingulate cortex and the prefrontal cortex. This part of the brain here is a driver for depression. And we've known now for about 20 years that many treatments of depression whether they're drugs like SSRIs, whether it's cognitive behavior therapy, whether it's ECT or sleep deprivation, even placebo. If you get better on placebo, activity in this part of the brain is reduced. And it seems that this part of the brain actually drives the negative thinking and also the some of the physiological changes like sleep disruption and depression. And it was this image here is an image of our research with psilocybin. And we, to our surprise, we didn't expect to see that psilocybin switched off this part of the brain. And that was one of the reasons we went on to do studies with psilocybin in depression. And I'll come back to that a bit later. So we've talked about the fast switch transmitters, the on-off switch, the GABA, glutamate. But what about those neuromodulators, dopamine, serotonin, noradrenaline, etc.? Well, these are hugely important because they, as I say, they add color and valence and flavor to what we do. And I'll just give you some examples of what serotonin, or 5-HT. Serotonin, there are 15 different serotonin receptors in the brain. So serotonin can turn on neurons through one, it can turn off neurons through others, it can modulate neurons through others. It has a multitude of different actions. And the reason it's got so many receptors is that evolutionary terms, it's a very, very old neurotransmitter system. And evolution has mutated or allowed receptors to mutate so that many different receptors now are sensitive to serotonin. And those receptors regulate mood, they regulate appetite, they regulate behavioral restraint in terms of controlling impulsivity, they regulate body temperature. It's a phenomenally broad set of behaviors and processes that are regulated by serotonin. Noradrenaline, a more recently evolved neurotransmitter that has a more limited repertoire, but that regulates important activities like arousal, attention, and blood pressure. And of course, those are kind of linked. That's the link of the heart and the blood pressure and the arousal of the brain are important because noradrenaline is a major transmitter in encoding emotional responses to stress and threat. And then there's the dopamine, the most recently evolved of the amine transmitters, and that's what we call the motive system now. That dopamine is involved in registering things that are important, whether they're good or bad, and therefore encoding salience and also directing activities towards either going towards the salient, a positive salience, or going away from a negative salience. And so that dopamine gives you energy and also, in the case of positive salience, pleasure. And many of the drugs we use and many of the drugs we take recreationally target these systems. So we've got antidepressants on the serotonin system. We've got MDMA on the serotonin system, releasing serotonin, enhancing serotonin. We've got psychedelics. I've shown you an image of psilocybin working through one of the serotonin receptors, dampening down the depression centers. LSD does the same thing. And migraine medicines also work on the serotonin system, but the serotonin system in blood vessels rather than the brain. Noradrenaline, we have drugs like beta blockers, which dampen down stress responses and cardiac responses in the periphery. And we have caffeine, which turns on noradrenaline, which is one of the reasons it wakes us up in the morning, gets us going. And the dopamine system is a target of stimulants like cocaine, amphetamine, but also alcohol. One of the reasons alcohol is such a good drug is that it doesn't just relax you through GABA, but it also turns on a bit of dopamine to make you energized. So it's a dual acting drug. And of course, we've got drugs which block dopamine, like the antipsychotics, which are useful to treat disorders where there's too much dopamine, like schizophrenia or mania. And I've already touched on this, but I just want to emphasize the fact that the brain is a collection of billions, hundreds of billion of pyramidal cells. And each of these pyramidal cells has these dendrites, and each one of these is a kind of computer. And so the brain does its computing in two dimensions. The primary computing is done through one of these areas with these processes called dendrites, which are organized in what we call cortical columns. A group of pyramidal cells is a little mini computer, and the first processing of vision or hearing or thinking is all done in these cortical columns, which orientate themselves at right angles to the surface of the brain, wherever you are looking in the brain, or certainly in the cortex. But then, of course, there has to be cross-cortical connection. And you look at the enormous size of the brain. you're connecting across centimeters of brain space between the visual system and the auditory system, and then the front of the brain, the thinking system. And that cross-cortical connections, the second level of computing, is absolutely crucial. Without it, we would just be a series of disconnected experiences. And this cross-cortical connection is done through these layer 5 pyramidal cells. And these are the ones which orchestrate and integrate brain functioning. And this phenomenal power of the human brain is largely pulled together by these special neurons in the layer 5 of the cortex. And I've said this already, the computing power of a human brain is more than all the computers on Earth today. Now, another really important construct is that the brain is not like a computer in the sense that it doesn't just replicate or copy what's out there. And the reason the brain is more efficient than all computers put together is because it works in a different way to computers. It works in a way which AI is trying to replicate. So for instance, let me give you an example of how the brain allows you to see things. You don't see what's out there. Your brain doesn't take a picture of what your eye is seeing. What happens is that photons from the world enter your eye. They activate neurons in the retina, which is actually an extension of the brain. And a lot of processing goes on in the retina to turn the colors, the movement, the intensity of the photons coming in into electrical impulses. Those electrical impulses pass up the optic nerve and they go into different parts of the visual cortex. So parts of the visual cortex detect color, other parts detect movement, other parts detect shape. And that your visual cortex, which is a large chunk, it may be 15–20% of your brain, reconstructs what your brain thinks is out there. So your brain has worked out already that I'm the person giving this lecture, which is why it was perfectly sensible for me to switch off the camera, because your brain had already worked out that I'm going to be me. As long as you're hearing me, then you're seeing me. So why bother to update what I look like? Because I won't change. I can't change. If I'd actually carried on being seen, if I turned into a polar bear, you wouldn't have noticed. Your brain just wouldn't believe it because it couldn't happen. So your brain is a hypothesis generating machine, and it's extremely good at it. And by doing that, it actually makes itself very efficient because you don't need to update. You only update when something changes. You don't update when nothing changes. But because your brain's very efficient, it doesn't generally care about very much. It cares about what you care about. In the context of today, it's caring largely about what you're seeing on the slides. And most of you aren't even taking any notice of what's going on in the room around you. And this is there's a wonderful quote which explains this from William Blake, the man who wrote the words to the song Jerusalem. He was a great mystic and a painter and a philosopher in a way. And he was also a neuroscientist, because he said this. He said, man sees through the chinks of his cavern. Basically, most of us have a very relatively limited perspective on the world. It's as if we're looking out from a cavern. But again, for most of us, when we look out through the chinks of our cavern, we do at least see a blue sky and pleasant white clouds. But because the brain constructs the images of what it sees, with people with depression, they often see horrible things. They see either blackness, or they see, sometimes in their dreams, they can even see souls burning in hell. Whereas addicts, when they look out for their cavern, they just see their love objects, the bottle or the syringe or the white powder. And that distortion of what's out there underpins many psychiatric disorders. And it was the writings of Blake that Aldous Huxley turned to explain his psychedelic experiences. So the reason he called his first book on psychedelics The Doors of Perception was because he was quoting William Blake. He realized that the experience that of his mind being opened by mescaline, which comes from the peyote cactus, and then he replicated it with LSD, could be best explained by the writings of Blake. And this is the quote from Blake: if the doors of perception were cleansed, everything would appear to man as it is, infinite. For man has closed himself up till he sees all things through narrow chinks of his cavern. And the great thing about Huxley was that he realized that psychedelics had cleansed the doors of perception in his mind. And then he asked the question, well, what is closing those doors? Or what is actually making them opaque? And of course, he came to the obvious conclusion, but it was a really brilliant insight. It's the brain. And he said the brain is an instrument for focusing the mind. And he realized that mescaline, psychedelics opened his mind because they unfocused the brain. And that is truly one of the great insights into the nature of the human experience, because we have now shown through our imaging studies that is exactly what psychedelics do. Psychedelics open up the brain, they increase connectivity in the brain, and they open up the mind, therefore. And on the left-hand side, this image here is called the small world brain, where most of the brain just talks to itself. So the visual cortex talks to the visual cortex, auditory cortex, auditory cortex. Of course, there's got to be some crosstalk. If your eyes see a bus bearing down on you, you've got to get your legs moving to run out of the way. But the efficiency of the brain comes by limiting the amount of crosstalk. Under psychedelics, pretty much everywhere in the brain can communicate with everywhere else. And that's how the brain was when you were a child or a baby. All things were possible, all connections could be made. And one of the major processes which goes on in development, child development, adult development, is that connectivity gradually gets whittled down till it becomes much, much more refined, much more limited in this small world construct. But the possibility doesn't disappear, and psychedelics bring it back. And that is why they're so powerful, because they allow people to see things differently in a way they haven't been allowed to, often for decades, because of education and learning and the processes of life. And by doing that, people can see new solutions to their problems, and psychedelics can help people escape from conditions like depression. And also that increased connectivity explains one of the most well discussed and most memorable for many people experiences of psychedelics, which are these powerful, complex hallucinations. People float out of their room, they float into a different universe. Many people go to mountains with white lights and they see God, and they have these powerful visual hallucinations. And that's because under psychedelics, the visual cortex, which normally talks just to the other, this is visual 1, this is visual 2, this is the central part of the visual cortex. Normally it doesn't bother to talk to anywhere in the brain except a little bit to the hippocampus for memory. But under LSD, with your eyes closed, your visual cortex is talking to all parts of your brain. So it's going back into your memory banks, and that's why people have these powerful visual hallucinations with the eyes closed. That it's like dreaming, but being awake. And to explain that, I've come up with this very simple two-dimensional theory of consciousness. I've already given you this one, the glutamate theory, which is about parcellation, about making things very distinct and precise. Whereas in the psychedelic stage, you have a completely different kind of sense of consciousness, which is complementary. You still remember what your experience is, but your experience, the content is different. It's not about parcellation, it's about integration. It's about being one with the whole, being one with the universe, being part of everything. And often the psychedelic state gives you valence, a sense of importance, and a sense of meaning as well. And that's through the stimulation of these serotonin 2A receptors, which are, as I say, I think mediate a different form of consciousness, which is complementary to and often additive with the conventional consciousness. And one of the other areas that has really developed in terms of understanding the brain has really come out of the of brain imaging, and that's the functional networks of the brain. So we kind of knew from studies of soldiers who had bits blown out that there were networks in the brain that did things, but now we can very explicitly interrogate them using imaging studies. This is data from fMRI, looking at which parts of the brain show activity that correlates with each other. And if there's a region where there's correlated activity, then it's a region, it's a functional region. So you see here, you've got the visual, the central visual and the lateral visual regions. So the core seeing, and then the more attributions of importance to vision. You've got the auditory system, you've got the sensory motor system here. You've got the executive control system at the front here. But you've got this region here, which is the most interesting one in many ways, the default mode. And if you just, the default mode is so called because it's that part of it's a network which is active when the other networks aren't active. So if you were to close your eyes and I was to shut up and I was to ask you just to think about whether this lecture's been worth coming to so far, then, and I was to scan you, I would discover that your default mode network, this frontal and posterior region, the posterior cingulate cortex, and these lateral parietal regions, this would be the network that was active while you were just reflecting on yourself and the value of decisions you'd made today to come and hear my lecture. And you see it's anti-correlated with the motor network. As soon as you were to wake up and open your eyes or move your fingers or touch yourself, then the default network would switch off because these other networks will become activated. But when you're just reflecting on yourself, the default mode is the driver. And that's because the default mode is where you encode your sense of self. And it turns out that psychedelics dramatically change that. And that's why they work in depression, because in depression, the self-referencing default mode network is overactive. More of the brain is consumed with self-referential thinking in depression, in depressed people here, than in people who aren't depressed. And that increased connectivity of the default mode is driven by this subgenual cingulate, the region that drives depression. And the greater the connectivity of this subgenual cingulate to the frontal regions, the greater the rumination. So we have a neurobiological circuit of depression, which is largely centered in the self-referential default mode network. Now it's negative, thinking is hostile and self-critical, but it can be all-consuming. And breaking down that network is one of the things that psychedelics do. So the reason people often feel that they've gone to a different dimension or floated into a different part of the universe is because their sense of self has become completely disrupted because the default mode network is completely disrupted. So here's the default mode network sideways on. Under psilocybin, this connection, it completely disappears. And that's why people often feel that they have floated out and they've gone somewhere else in the universe. Their body has morphed or atomized. And it's that disruption of the default mode network we think explains the powerful effect of psilocybin, a single dose of psilocybin, to reduce depression in people with resistant depression. The most powerful treatment of resistant depression there's ever been. And it's because we can break down the repetitive thinking processes which underpin depression. I want to finish just by talking about another kind of imaging, which is one I'm particularly fascinated by, because it allows us to measure chemistry and chemicals in the brain, and it's called PET scanning. And here we can measure the density of receptors in the brain for neurotransmitters, and we can measure the release of neurotransmitters in the brain. And that's something I've spent the last 25 years of my life trying to do. And we now have tracers which can allow us to look at a whole series of different receptors and neurotransmitters in the brain. We can look at brain metabolism. We can look at GABA receptors. We can look at dopamine transporters. We can look at dopamine receptors. We look at NMDA receptors. We can look at serotonin. We can look at a range of different neurotransmitters and receptors in the brain. And this can be really important in helping understand why, for instance, the brain gets dysregulated in depression. And that is simply, in simple terms, because the inhibitory serotonin receptors, or serotonin 1A receptors, which are labeled by this tracer, which are centered in the limbic system here, they're downregulated in depression. Depressed people have reduced density of these receptors in areas of the brain like the default mode, the anterior cingulate cortex. And these are inhibitory receptors. And it may be the reason you have this excessive activity of the default mode is because the serotonin system isn't inhibiting it appropriately. So this is one powerful visualization of a neurochemical abnormality which could explain depression. And we can also come up with similar theories relating to anxiety disorders. There's the Edvard Munch, the scream, which many people interpret as being a panic attack. And we showed years ago that people with prone to panic attacks have a deficiency of GABA receptors in their brain. And without those GABA receptors, they struggle to restrain anxiety. So one of the reasons why drugs like benzos and also alcohol can reduce anxiety in anxious people is because they're replenishing a deficient GABA system in the brain. So this kind of imaging is hugely powerful, gives you hugely powerful explanations as to some aspects of brain dysfunction in disorders like depression and anxiety. So I'll stop now. We've got about 30 odd minutes for questions. This is the book that has just come out, and you probably all know about Drugs Are the Hot Air. I don't know if you all know about my autobiography. If you want to read more about, particularly about why we got into psychedelic research and research with MDMA, and also my battles with the government, you can buy my autobiography, and they all make wonderful Christmas presents, particularly for your parents. So feel free to buy them, and also remind you that all the proceeds support my charity, Drug Science. Thank you very much.
That was brilliant. Thank you so much. I feel like you've just covered all of my sort of degree in neuroscience in about an hour or 40 minutes or however it was.
Well, I've made it simpler anyway.
But it was fantastic, and we've got lots and lots of questions. So I'm very sorry to anyone whose questions we don't get through, but we will try our best. So the first one that's popped up is, what is the mechanism for memory loss in depression? Which I thought was very interesting.
Yes, well, it's an interesting question. I mean, up till recently, there's been a lot of talk about neurotrophic problems. The fact that in depression you deplete the levels of a neurotrophic hormone called BDNF, brain-derived neurotrophic factor, which allows the pyramidal cells to function normally, and that's actually one very plausible explanation, that if you need the BDNF to keep the massive branching of those pyramidal cells, if you deplete BDNF, certainly in animals, the branching gets less and the number of synapses get less. So you actually reduce the computing power of each pyramidal cell, and therefore you reduce the computing power of the brain. So that was the theory that I was propounding, until relatively recently, and I think it's still true. But I think there's another angle, and that is this overactivity of the default mode. Now, having kind of discovered this and from other people's work, it kind of makes sense, because if you're using, as you saw from those images, I mean, I don't know if can I still share? I can share. Yeah, I can probably share, can't I? Where's that? Where's he gone? Where's my talk gone? Here. Have you come back? Here he comes. Yeah, I mean, if you've got 3 times as much of your brain engaged in self-referential thinking, you've got a lot less of it available to do other kind of things. So you can certainly see why executive function, which comes from this part of the brain, might be downgraded in depression, because a lot of the part of the brain that's trying to do executive function and thinking and memory is actually engaged in thinking about yourself. So it's like the brain has shifted away from externalizing and allowing you to, or even access positive memories. It's become completely obsessed, and large chunks of it are just engaged with recycling these negative memories. And that touches on really what I think is perhaps the most critical question of all, which I don't have an answer to, and I want to try to work on at present, is why would you switch? I mean, it's clearly maladaptive to switch to this state, spend so much of your brain power thinking these negative thoughts. What is driving that? And we know it's this region here, but why would this region suddenly take over the brain? What's going on there? And the answer is we don't really know. I mean, one theory is there is a deficiency of serotonin there, and serotonin is a switch, and there is some evidence for that. Another possibility is that somehow glutamate there becomes overactive and drives the system inappropriately. But why you should have such a complex negative network of negative thoughts, it tells you something. It tells you that you don't forget memories. Bad memories are in your brain, even though they're not generally interfering with your life. But if they do come into consciousness, they can dominate us in depression.
I always think it's amazing that you can sort of remember that time that you were 5 years old and you dropped your tray in front of the entire preschool or whatever. But I can't remember what I should have learned last week.
Yes. Well, that's right. There's encoding, encoding negative experiences which are potentially damaging is quite a useful thing. It is useful to remember. I do remember vividly falling backwards sitting on a fire when I was about 8. That was very painful, and I've always been very cautious about turning my back on fires since.
Yeah, I bet. I'm not doing that again. Gosh, yeah. That's very interesting. I knew sort of about the neuronal atrophy thing, but I'd never thought about memory loss in terms of the default mode network.
So it's not lost. It's basically it'll come back. The memory, if you get rid of if you get the default mode sorted, and also you can also restore the BDNF by antidepressants, for instance, then it'll come back. And there is actually, just to finish with this, one of the interesting things about psychedelics in depression is they're also neurotrophic. So they may even more actively restore those synaptic branches and the synaptic spines.
That's very, very interesting. And ketamine is they've done similar.
That's the same, absolutely. Ketamine increases connectivity and increases BDNF.
Yeah, it's very interesting. I love this. One of my favorite topics. On that same sort of train of thought, there's a question here, I think, about how psilocybin maintains new connections and enhance creativity, and for how long does it work in quieting the default mode network and reduce ruminating thoughts and things?
Well, so it does that for the duration of the trip. Okay. But then afterwards, there is less connectivity between the default mode network. So we haven't kind of clinched that, but our belief is that two things happen, is that you disrupt particularly the emotional circuit. You get more control over the access to the negative memories, and that's probably through some kind of prefrontal some disruption of that of the process, which allows the prefrontal cortex to get to re-establish control over the amygdala, where and the subgenual cingulate, where these negative memories are being driven from. So that's the first thing. But the second thing is that, and this is possibly more important, is that we know, and we haven't published this yet, but it's under review and hopefully it'll come out fairly soon. We know that brain flexibility after psychedelics is enhanced, and it stays enhanced for months after. And we think that flexibility protects you against getting locked into ruminative thoughts. It allows you to think in a more flexible way. So what sort of working hypothesis then is that we can disrupt depression. People can escape from depression. They can have this wonderful experience of making connections which they hadn't made before, connections which will allow them to understand why they've got depressed, but also to come up with new solutions to new ways of interpreting and analyzing those experiences and those memories, and plot new ways of thinking. And also basically people often come out and say, I'm going to be different. I want to be different. I'm going to be like, and the final thing is that the neuroplasticity promoting effect of psychedelics can actually then help you learn to keep staying, to hold that moment of insight and maintain it as a way of thinking in perpetuity. So there is a possibility you can actually, and some people, you may get a cure. Let me just go and talk a bit more about this in the context of the depression trial, which was here. So here you see some of these people they're well 8 years later. So for some people, the single dose of psychedelic put their brain back in a non-depressed state. But for the majority, although the effect was huge to start with, it wore off. And this, I think, is testimony to the fact that depression is a peculiar brain state, which is it's a kind of overlearned brain state. I think for people, particularly people whose depression starts in childhood due to neglect or trauma, it may be actually the normal state of the brain. The negative thinking may just be what you are. So getting rid of that might require repeated administration. So eventually we might be able to kind of push it down so it doesn't intrude into consciousness eventually. But that's very much a project that needs to be done.
God, it's fascinating. I'm glad you showed us the slide again. And there's a few questions sort of about more structural stuff of the brain. So there's one that's asking, as well as the vertical anatomy of the brain, do you see significance in hemisphere lateralization?
Well, yeah, I mean, clearly it's extraordinarily important. I mean, it's very important in language, but it's more important, there's clearly lateralization in high level cognitive thinking, and there's also a degree of lateralization also in emotion. Whether you can use lateralization, I mean, it's always a problem. I mean, lateralization clearly is fascinating, but and you can, you can study it now. You can dampen that. You can zap one side of the brain and show you can distort the visual pathways, etc. Whether it's it's a fascinating phenomenon, probably driven to a significant extent by the lateralization of language, which we still don't understand, but or we don't understand the reason for it. But whether it's got beyond being a fascinating issue, I'm not sure it's got a lot of meaning in terms of explaining how the brain works. It's just that different bits of the brain do different things. But we knew that anyway. The visual cortex is structurally very similar to the auditory cortex. It's just that one does vision, one does hearing. So one hemisphere does more abstract constructs and one does more concrete semantic constructs.
Yeah, it's very interesting. I actually I remember reading, and I think it all comes down to plasticity, but the brain that changes itself, which is a good book about sort of plasticity, I suppose. Before I started my degree, and there's a story about a young boy who's in a car accident and really damages one side of his brain, probably wouldn't survive it as an adult, and so his all his centers switched, but the things that you learned first as a child filled in whatever brain space was available, and then sort of those other functions like arithmetic that came later he could never do. And I always thought it was a very interesting kind of example of like, yes, we have lateralization, but actually it's not necessarily that important in a way. I mean, it's important, but not that
no.
important.
And another example, no, because I mean, one of the most remarkable things that's happening, I mean, you've seen these new data on how you can put vibrations into the skin and get people who are deaf to hear. Now that is fascinating, because that tells you that the auditory cortex and the sensory cortex can in the end, and I don't know whether it does connect to the auditory cortex, or whether you hear in your actually I don't know enough about it to know whether you hear actually in the sensory cortex, but the nature of sound can be communicated through stimulating touch receptors. So there are these cortical columns have a remarkable capacity. So the visual cortical column can be in a blind person can be adjusted to become an auditory column. So they have that kind of flex. So it is a bit like a computer. It's not it's about the program you put into it, which gives you the output, not the hardware.
Yeah, I mean, and there's a question here, which I think you've probably sort of answered, but is there a structural difference in the connectivity of the neurons in different areas of the brain? So why does one set of neurons firing result in color, another sound, another is emotion?
Well, that's learning. I think, and we know that. And it's the message is really important, is that if you haven't got exposure, you can't learn. And that's why rich environments for children, that's why the Romanian orphans aren't very clever and actually have quite a lot of problems in relation to even socialization as well as intellectual development. Because if you don't basically allow the brain to optimize its responsivity, you end up failing to achieve its full potential. So the lesson is absolutely clear that children need to have rich, interesting environments that are sound and vision and tactile. In many areas of life we probably are already denying. I mean, the tactility. COVID has actually had a very negative impact on tactile responses. So we need all those things, but on top of that, it's also really important. This is one thing that I've got more and more convinced by our data and depression. It's so important that children are brought up in a completely positive, emotionally supportive environment. It seems like the brain is so easy for people to get to be responsive to and get locked into negative thoughts. And see, I never thought I'd be saying this. I was brought up interested in Freud as a kind of construct, but not believing that Freudian concepts really had a lot of utility in terms of, say, neuroscience or in terms of psychiatry. But now more and more I'm thinking of certainly Klein, Freud's daughter, in terms of the nature of childhood. Young children believe, well, the Kleinian view is that young children believe that they are the center of the universe, that they are the most important thing in the world, that they're omnipotent, and the mothers serve them. And then gradually they learn that they're not. But the problem is that sense of omnipotence, is really important, because if they discover very early on that they're actually not omnipotent and that they're hated or abused by their parents, because they still think they're the cause of everything, they blame themselves. So you can see that you can basically develop a mindset as a child that you are the problem. You cause your parents' divorce. You deserve to be abused. And if you set that in the first few years of life, it can be extremely difficult to break. So now I'm very concerned with the fact that I want, hopefully our evidence will support the view that we should really be investing in child development and trying to minimize any kind of serious harm that comes to children, protecting children from the worst excesses, often of their own parents.
That's really interesting. And I suppose, as soon as you said that, you think about, we understand that there's a way that children learn and they need input for it, and how formative that is in forming their brain structure, and like critical periods for vision. If children can't see before the age of 5 for various reasons, they don't get input, then that doesn't form. So of course that emotional input is going to have just as much as a positive or damaging.
Well, I fear that negative inputs may be even more powerfully destructive. I think positive inputs are, it may take a lot of positivity to offset one really nasty negative. It's just, it's I don't know, but anyway, whatever we do, the more pleasure and the more happiness we can give to children, I think the more likely they are to grow up being, content and positive people.
No, it's really, really interesting. And moving from children to computers, because I've seen a few questions based around computers. So I think there's two parts to this. So I think one is asking your sort of opinion on whether AI is going to catch us up or supplant us. And the other is just asking if, we have such a human powerful brain, but why can computers do much better maths than we can?
Yeah, well, the second one's easy to answer because they were built to do maths.
Anything else?
Yeah, that's right. The human brain was built largely to make human society, which is infinitely more interesting and complex than a computer. Humans make computers, not the other way around. Let's get real about this. What's adding up? Come on, guys. That's just a boring thing. Computers won't invent the Taj Mahal or they won't paint the Sistine Chapel, because that's not what they're built to do. I mean, eventually, of course, there are computers now that can make music, and that's, they can mimic what we do if we program them right. In terms of AI, well, yes, I'm like a Hawkingite here. I think he's right. And in the end, robots will take over, because we're so incompetent at dealing with we're very creative, but we're also stupid and selfish as a species. So I am a bit pessimistic. It's the only way that won't happen is if we destroy the world with our atomic arsenals. But eventually, I guess computers will do, robots will, eventually do so much that they will probably, they will eventually gain autonomy. It's hard to imagine how they couldn't unless we actively kind of castrated them at birth. But it won't be in my lifetime. Might not be in yours either, Anna. We might have another couple of generations while we're still able to destroy the world as we're doing now, and then it'll either be robots or cockroaches will take over.
Oh no, I know. Well, I mean, my saving from this is I always remember learning in neuroscience about how automatically we catch something when it's coming towards us, and how we are calculating all the time as an object comes towards you and things, and you just catch it and you don't even think about it, unless you're very bad at catching, in which case it is.
Well, if you think about it, you're going to fail.
Yeah, well, that's true. Well, exactly. But to program a robot arm to do that, to catch kind of any object is still almost impossible, is my understanding. I'm happy about that.
Indeed. Yeah, no, I quite. That's. I think it's important. Well, there are the bigger questions. We've delegated so much decision making now to computers. It's one thing for sure. If someone pulled the plug on all of them, human life as we know would just pretty much disappear. At the very least, we start communicating through Zoom, wouldn't we?
I know. What would we do during another pandemic?
What would we do? Exactly. Yeah.
There was also, I thought, a very interesting question about whether mushrooms that contain psilocybin whether you think they'd have grown had we not had 5-HT receptors in our brain.
Yeah, no, the mushroom predates serotonin, predates humans. I mean, mushroom. So yeah, the interest. Why do mushrooms have psilocybin? It's definitely not for our pleasure. Well, some people believe that. I mean, some people think mushrooms are seeded on Earth by aliens so that human consciousness could expand to the point where it made the Earth a better place. I mean, that's Hancock's view. I mean, yeah, it's an entertaining theory. I think it's unlikely, but I can't prove it's wrong. I think, why are they there? Well, they may facilitate dissemination of spores because animals eat them, or they might be deterrent, or they're probably a bit of both, to be honest. They're probably a deterrent to bacteria eating them, and probably have some positive effect on other animals eating them and dispersing them.
Yeah.
So what I think, I mean, chemicals in plants usually have that, either one of those two functions, usually to stop insects eating them, or to encourage animals to eat them. So one of the theories as to why fruits, nuts, grow like plums, or, have a big juicy outside, is to encourage animals to eat them, swallow the kernel, wander off, and poop it out. There's a new fruit tree. Yeah.
It's a circle of life.
Yeah.
There's also a question about do we know why sleep occurs less as we age, and why those sleep cycles change?
No, well, yeah, we don't. It's aging. We actually don't know. I think it's just generally that most things get worse as you age. Certainly your memory gets worse, your processing speed gets worse, your hearing gets worse, your vision. We know why those are. That's cataracts and things. But no, actually, we don't generally know. We don't. We don't. Or at least if we do, I don't.
I mean, I think sleep generally is still a surprising mystery. We can measure it quite well, but as to understanding much more.
Yeah. Well, it's quite complicated because there are 4 or 5 neurotransmitters which promote sleep, and about 7 or 8 which keep you awake. So it's actually a very complicated equation. It doesn't, and we know, for instance, with disorders like narcolepsy, you can change just one of those arousal promoting systems and get problems with sleep, with arousal. So it's probably the same the other way. You probably begin to get slight attrition in the sleep promoting centers, and they sort of magnify the effects, perhaps because there's some sort of reciprocation there. But I genuinely don't know. We don't know why people.
No.
We don't.
It's baffling. And sort of going from sleep, we're going into, someone's got a question about comatose states, which is, could you explain the functions of the brain and mind as far as the ability to hear and feel go when someone is perhaps comatose or maybe locked in syndrome?
You're going to have to eat in the next 5 minutes if I'm going to be zooming in the kitchen, and you need to dish up your food.
I think we'll be wrapping up very shortly.
Sorry, sorry, just organising the complexity of having 3 separate people on Zooms in the Zoom meetings one evening.
I'm very lucky. I've set this. I've been given this room for the evening. I'm safe.
So, sorry. The question was comatose states. Yes.
Yeah.
So, what was it? What was the question?
How can people sort of hear and feel and things? I think maybe locked-in syndrome might be.
Locked-in syndrome. Yeah, well, that's really interesting. And that's because, well, the simplest explanation is that you've got the human brain, which is the cortex is impaired, but you've definitely got the lower brain regions working, which is, of course, obviously they have to be, otherwise you wouldn't be breathing and eating. And there's also the emotional centers to some extent which are functioning. So yeah, you can clearly, there are bits of the brain which are working now. One of the really interesting questions is that, which you touched on that previously, is that the arousal systems in the brain are, there are multiples of them, but even just if you perturb one of them, like the orexin system, you can get weird states like cataplexy. If you disturb 2 or 3 of the arousal systems, like the noradrenaline, dopamine one, you can get into a state, a kind of comatose state, where it's actually where you can't have volitional activity in terms of motor activity, which is what most people think about as WhatsApp, but you can clearly have some thinking. And well, it's a bit like dreaming, your brain is very active in dreaming, but you're not moving. So some aspects of locked-in states are due to failure of upward arousal systems. But I'm quite interested in the opposite. There are some conditions where people are locked in because the cortex is overactive. So rather than being overactive in causing depressive thinking, it's overactive in suppressing subcortical activity. So some locked-in states are due to the brain imposing suppression on activity. And the reason we know that is because there are drugs we can use like zolpidem, which largely are cortical. You can switch off the cortex with zolpidem, and people can move, can come out of these locked-in states to some extent, and certainly can eat better. So that's kind of proof that there are states of cortical overactivity. And in fact, we did some nice experiments 15 years ago. We showed that if you dampen down the cortex with zolpidem, you actually have more activity in the subcortical centers. So in comparison with other sedative drugs which dampens them more down. So we could actually show that, you can disrupt the cortical-subcortical balance by just targeting a drug on the cortex.
That's so interesting. I actually had never really thought about the fact that you could sort of have locked in from damage or from one system, or actually just overactivity.
Well, yeah, I mean, you can also, but you've obviously seen it in conditions like hypnosis. I mean, hypnosis is a state which is largely some kind of cortical suppression of motor activity. People can do very strange things under hypnosis, a bit like, they can if they're in kind of strange states of cataplexy and psychosis.
So I think probably have time for one more. Last one.
Would be great.
7:30, and I've just lost it. Oh yeah, it was just which I think is a very interesting question, but asking about how psilocybin interacts with the brain, and do we think it's just due to sort of 5-HT2A receptors, or are there other neurotransmitters that it's interacting with?
No, it's 2A. We know that because you can block the 2A receptor and you can actually stop all these psychedelic effects of psilocybin. Yeah. Right. I'm going to have to leave you now.
Yeah.
I'm vacating the office. My wife is going to be doing it, and I'm going to go and eat. Great to talk to you all.
Thank you so much. Thank you. Bye bye. And I will just wrap up. Thank you all so much for coming. I know that we didn't get to go through all the questions, but a lot of them, I know, having read about 70% of this book, can be found in here. And it does make good Christmas presents, because that is also what I am giving to my mum. But yeah, I hope you all have a lovely winter and holiday, and I hope you learned lots. And that's probably everything from me. And thank you to David, even though he's gone.